it's going to synthesize lipids for membrane growth and repair. So let's say I have a piece of the cell membrane all the way over here. If I have a little bubble of lipids that can bump into it, eventually those lipids will fuse with the cell membrane and become a part of it. And in a series of steps, I can go from here to fusing to opening up, and the cell membrane can grow a little bit. And conversely, the opposite thing can be true. Sometimes we can pinch in a bubble of membrane and bring it in if the cell has some damaged membrane. So our cells are rather dynamic. The membrane is growing and contracting, or at least removing pieces that are damaged and replacing it. There's always membrane growth and repair going on, and the smooth endoplasmic reticulum plays an important role in membrane growth and repair by synthesizing lipids and carbohydrates for cell membrane. So I hope you got the difference between smooth ER and rough ER. Think about that. Now, I want to try to wrap this video up before it gets too long, so let me finish the last few organelles, and I'm going to stop, and I'm going to do a second video called The Cell Story, which is written in the note set. If you're in my class and you have the note set, I'll tell you what page it's on when the time comes. So, the last couple of organelles we need to talk about. There's the Golgi apparatus, there's mitochondria, and there's lysosomes. So let's talk about them. For the Golgi apparatus goes by several names. It can also be called the Golgi body or the Golgi complex. Different books use different names for it. Golgi body, Golgi apparatus, Golgi complex. This is a series of flattened membranous sacs. They contain enzymes. Remember, enzymes from a previous lecture are proteins that are involved in biochemical reactions that alter proteins for secretion. Now, again, this is a rather very simplified view of these things. But when I look inside the cell, if I have some endoplasmic reticulum over here, and let's say it's rough endoplasmic reticulum, it's got ribosomes all over the surface, and we've pinched off a transport vesicle from this piece of protein, from this piece of endoplasmic reticulum. This transport vesicle has in it a number of different proteins. And let's say these little proteins all look like this. As we transport this vesicle, and we're going to try to secrete these proteins, let's say this is the cell membrane out here. Before we secrete these proteins, we better make sure that they're functional and they're in the right form. To me, the Golgi apparatus, when we're shipping proteins out of the cell, is almost like quality control in a business. I tend to think of the cell as a factory. It's a factory that makes proteins, for the most part, as products. The factory workers are the ribosomes. The instructions are in the president's office called the nucleus on how to make all the products. If you're going to ship a protein, you don't just stick a stamp on it and send it off. If you were going to send your laptop to a cousin in Minnesota, you don't stick a stamp on it and drop it in the mailbox. It will disappear and it won't make it. So you put it in a package. This is our transport vesicle. You're going to transport the package, but before we send it out of our factory, we have these little disc-like proteins, disc-like membranes. If I slice one open like this, it's a little disc-like sac of membrane, almost like little pancakes, so to speak. And so I have this series of flattened membranous stacks, like a little stack of pancakes. They're all stacked on top of each other like this in 3 dimensions. And we're looking at it from this funky angle, okay? Now, because it's made out of the same membranes as this transport vesicle, before I secrete these proteins, this transport vesicle can come over and fuse with the Golgi apparatus and put the proteins inside of it. And let's say the proteins need to be modified or altered for some reason, then when it passes through the series of, oops, that marker doesn't work. When it passes through the series of membranous sacs and comes out the last one, we can pinch a little bubble of membrane off of this that will have the protein in it, but now the protein might be altered to some degree. And you can't see that marker, so I need to get rid of it. But the protein can be altered. Now when this bubble of membrane fuses with the cell membrane, the protein is released outside the cell. It has been secreted. So there's a thing called protein sorting, and when proteins are going to be secreted, they usually go through the rough endoplasmic reticulum. They get shipped by a transport vesicle to the Golgi apparatus. The Golgi apparatus will make sure the protein is functional and proper. If not, it can modify it or correct it or alter it to some degree, and then it packages the protein in what we call a secretory vesicle. And a secretory vesicle is simply a vesicle that's going to fuse with the cell membrane and secrete the protein. So that's the function of the Golgi apparatus. It has enzymes that can alter proteins that are going to be secreted. If you have a factory that orders, or a business that orders something from you, and you don't send them the right product, what's the likelihood your business stays in business? It doesn't. People will stop using you, it will crash, and it will fold. The same is true for the cell. The cells are dependent upon each other, and some cells sometimes need a protein that this cell makes that they cannot. And so this cell can release this protein into the extracellular fluid, and it can go and be transported somewhere else. I hope you're getting a pretty good picture of how the cell works. Now let me take all of this off the board, and we have to do two more organelles that I want to discuss, okay? And I think we're going to be past the limit, so this video will probably time out, and it automatically starts another one, and there'll be two parts to this video, probably online, okay? So now, mitochondria. And if you see it written this way, mitochondrion, that's one. Mitochondria is two or more. It's plural for mitochondrion. They're the same thing. So what mitochondria do is this. They contain enzymes that convert glucose and oxygen into ATP, which is the cellular energy. Mitochondria are these little organelles, and they actually have two membranes. They have an outer membrane, and then their inner membrane is all folded up. And inside this inner membrane and the fluid inside them, called the matrix, they have the ability to take some of the products from glucose and oxygen and turn them into energy. So here's a short synopsis. In our cytoplasm, with enzymes, I can convert glucose into 2 ATP. There's some byproducts that come off that go through here in the presence of oxygen and give me another 34 ATP. When I do the math, that's 36 ATP. I can convert glucose into ATP in the cytoplasm without mitochondria, but this is not enough energy. This would be like your car getting 2 blocks to a gallon. If you had to take a long trip, you would run out of gas or energy before you made it to the next fueling station. The beauty of mitochondria is they can take some of that glucose or the leftovers from the conversion of glucose into energy and oxygen and make a lot more ATP. So the way I always say it is that mitochondria convert glucose and oxygen into 36 ATP. That's not 100% accurate, but we'll get to that when we do cellular metabolism in the future, and we'll look at the details, okay? So they produce an overwhelming majority of the energy for the cell. That's what mitochondria do. In my opinion, if we were talking about this factory example, then mitochondria are the power plant that powers your factory. The last organelle we're going to talk about is called a lysosome. We could talk about peroxisomes and others, but I'm not going to. Anytime you see lys or lyso, it means to digest or break. So, and the some means body. So these are little digestive bodies. Now this is how we describe lysosomes. They are specialized vesicles that contain enzymes that digest foreign substances. They're little bubbles of membrane, specialized vesicles, that are filled with enzymes called digestive or lytic enzymes that can digest or lyse foreign substances that get inside the cell. Okay? I think of lysosomes as sort of the custodians and the security force of your cell. Okay? Hold on a second. Let me check something. Yeah. So, if I were to try to do the cell story right now, which I'm going to do, you'll see all these organelles come together. Now, your cell is like a factory. And if I look at the cell, the outer perimeter of our cell is actually a double membrane of lipids. That would be our cell membrane. Substances from outside the cell cannot get in, and substances inside the cell cannot get out without permeating or passing through the cell membrane. So it does the isolation, protection, permeability, and sensitivity. Usually somewhere near the middle of the cell is this organelle called the nucleus that would be like a three-dimensional little bubble, but it has these holes in it called nuclear pores. Inside the nucleus are the instructions of this stuff called DNA, Deoxyribonucleic Acid. And I told you before, the deoxyribonucleic acid, or the DNA, is the instructions on how to assemble every protein inside the cell. And it doesn't come in one large volume. Imagine a stack of encyclopedias. If you wanted to make proteins that have the letter A as their starting name, then you would have to go to the A volume to find their instructions. If I stuck the encyclopedia set into one giant book, it would be unmanageable. You'd have to pull it off a shelf like this, set it on a table, climb up on. Doesn't work. So it's broken into smaller volumes. Each volume of the encyclopedia set would be considered a chromosome for the entire genome or genetic makeup of the cell. So if I need to make a protein that starts with the letter A, I go to the A chromosome, or the first chromosome. If I need one that starts with the letter M, I go to the M volume, and I can find all the instructions for all of those proteins. Similar to that concept. So now, if I need to make a specific protein, all of the instructions are found inside the nucleus in the DNA. So the nucleus is protecting and housing and storing the DNA. If there's a section on a chromosome, a particular page that has the instructions for a protein that we need, I can go in there. Now, one of the things that happens is these little guys that are out in the cytoplasm, and I'm going to draw them with two little parts of their body, would be called the ribosomes. They have large and small subunits. We could get into that cell stuff, but it's not important. The ribosomes are just waiting for instructions to assemble a protein. If I need a protein that is found on this particular part of that chromosome, I can rewrite the DNA into messenger RNA. And the reason you will see is that the ribosomes are incapable of reading the alphabet of DNA. That's not going to make perfect sense to you yet, but it will. It would be like me having instructions here, maybe in Russian, but I can't read Russian. So someone would have to rewrite it or translate it. And I don't need all of the entire set of encyclopedia. I just need the instructions for that one protein rewritten from one language to another, from DNA to messenger RNA. When that mRNA comes out of the nuclear pore, the ribosome gets it, it will start to synthesize the protein by reading the instructions. This amino acid goes first, this amino acid goes second, and so on and so forth. If the protein is going to stay inside the cell, it will be synthesized on a free ribosome. When the ribosome is done, it releases the protein to do its job, it releases the instructions, and it goes on, and the instructions get degraded. It's not important to us right now. If the protein is going to be secreted, then there is a long series of interconnected membranous tubules called the Endoplasmic Reticulum that goes all over the cell. And it's a series of interconnected membranous tubules and I'll have sections of it everywhere. When you look through an electron microscope, in cells that are specialized for secretion, they're filled with endoplasmic reticulum. And if these ribosomes need to secrete this protein, then they're going to attach to the cell membrane in these spots and make it rough endoplasmic reticulum. As they synthesize their proteins, they'll shove all those little proteins inside the endoplasmic reticulum. The endoplasmic reticulum will pinch off little bubbles of membrane called transport vesicles that are going to transport these proteins. If the smooth endoplasmic reticulum pinches off a bubble of membrane, and by the way, it's going to have a double membrane just like the cell membrane. When this vesicle bumps in here, it can fuse with the cell membrane, and we get some membrane growth or repair from the smooth endoplasmic reticulum. That's why we say it synthesizes lipids and carbohydrates for membrane growth and repair. We're interested more in these transport vesicles that are going to transport proteins. And what they're going to transport them to is this little series of membranous sacs called the Golgi apparatus. As the transport vesicles move to the Golgi apparatus, they will fuse with it, and the proteins that they're making will be stuck inside the Golgi apparatus. The Golgi apparatus, like quality control, will start to modify the proteins if it's necessary. And by the time they get transferred through the various levels of the Golgi apparatus, when they come out the other end, the proteins might look slightly different. They might be modified. This is now going to be called a secretory vesicle. We've gone from a transport vesicle, which transport proteins from the rough endoplasmic reticulum to the Golgi apparatus, to a secretory vesicle, because this vesicle is going to bump into the cell membrane the same way the others do, and it's going to release those proteins or secrete those proteins into the extracellular fluid. So really my cell is a factory for proteins. The builders are the ribosomes, the instructions are the DNA or the nucleus. The ribosomes get a message called the messenger RNA. They will either synthesize a protein and release it into the cytoplasm if it's a free ribosome, or they will become bound to the endoplasmic reticulum. They will build the protein, and as they build the protein like this, they will shove this completed protein into the endoplasmic reticulum, this long tube of membrane. And then what they'll do is they'll pinch off a bubble of membrane called a transport vesicle that has the protein in it. When the Golgi apparatus receives the protein, it can check it and say, oh wait, that's not quite correct. I don't need that amino acid. I need this one. Oh, and by the way, not only can I alter it, it might add a little sugar or something here to it called a glycoprotein, not important. And then it will send that off into a secretory vesicle, and that will fuse with the cell membrane and secrete the protein. That's essentially what our cells are doing. They're little protein factories producing the proteins that are necessary for the processes of life to occur. Let me fix this. Now, along the way, there are these little organelles, and our books always use an orange color for them for some reason. Let me see if this orange marker shows up. There's little organelles called mitochondria that are going to convert glucose plus oxygen into ATP. They're making energy. So the mitochondria are producing the energy that allows all these processes to occur. And finally, the last one we talked about were these little guys called lysosomes. Sometimes the proteins that get pinched off here get pinched off into special vesicles that have proteins that can digest stuff. So let's say some foreign substance bumps into our cell. Some bad guy. The cell can actually invaginate or start to pull in its membrane like this. And as it does that, it can take in a bunch of these bad guys and pinch it into a little vesicle. If I fuse this vesicle with that one, these enzymes, as these two vesicles get fused together, these enzymes will destroy that foreign substance, literally break it down or digest it, and then we can go and release that stuff out of the cell like trash. That's actually how our immune system works. A lot of our white blood cells have lots of these lysosomes that can digest and break down lots of things. That's one way that they work. But all of our cells have these special lysosomes that contain enzymes that digest foreign substances that get into the cell. So now, if you go into my note set, we've listed all the organelles on page 14. We've discussed their functioning. You're going to have to skip quite a few pages. But if you flip to page 23, the cell story is written here. Everything that I've described and discussed with you is written here. And this is one of the last things in the note set before the exam, and right after that are actually 10 real sample questions that I wrote out from a real test. We'll go over those one day as a brief review before the first exam. But please read the cell story. Please memorize the functions of each one of the organelles, but be able to explain it to a 7th grader, and hopefully they will understand how all the organelles function to keep your cells alive. Again, this is a simplified and brief description. It would take 2 or 3 semesters of cell and molecular biology to really get into the details, and I'm not interested in that yet. We will add complexity as time goes on. Now look, this is a long video. It's probably going to be broken into 2 30-minute videos. I hope my camera did that, and I hope you enjoyed watching it. I hope you had as much fun as I did learning this stuff, and I hope to see you in the next video. Okay? Study hard, study often, do it till you can't stand it, do it till you understand it and can explain it to someone else. See ya.